Hello, and welcome to my next live stream. Firstly, as usual, can you just tell me sound quality and levels are fine and the visual quality is fine? Okay, firstly, I've been asked some questions by email, which I'm going to answer first, and there's some already in the chat, which I'll then answer, and only then can you start sending in questions. Can I do the Simpsons theme tune? No. I can play a bit of it on the keyboard, but no. Right. Firstly, I was just asked to do nucleophilic substitution with alcohols. So if we have, let's say, bromomethane, and we want to turn it to methanol. Now, you remember dipoles, carbon is slightly positive, bromine is slightly negative because the bromine is going to attract the electrons slightly more than the carbon. Curly arrow going to the carbon, from the bond there, one from the bond to the bromine, and then you have that's it. It's reasonably simple. Yeah, it's reasonably simple mechanism. So just curly arrow from the double bond lone pair on the OH minus group goes to the positive carbon, then the bond between the carbon and the bromine break, and you've got the electrons going to the bromine, which is slightly negative, and you've got the substitution there. I hope that makes sense. Next, I was asked just to go through alcohols on a NMR spectrum. So I've done one already. Here's one I prepared earlier. And essentially, if you're looking at an alcohol, the different types of bonds you have is a C to H group. So let's say I'm just going to draw methanol in this case, just so you can see. Actually, now I'll draw ethanol. So you have these H groups. You then have a carbon to carbon group. You have a carbon to oxygen. You have a carbon to hydrogen. So that's ethanol. This one here is propanol. I'll show ethanol because it fits. And then what you do is just look for the peaks. A C to O bond is, where's my formula sheet? C to O bond is shown by a sharp peak at 1,000 to 1,300. So we've clearly got that there. Now you'll always have a CH group at about 3,000, but I've drawn that in anyway. And the more important one is if there is a broad peak at 3,000 to 3,300, you have the OH group of an alcohol. That's all you need to know. For the alcohols, the other key one is if you see a sharp peak here, about 1,750, you have a CO group, which will be a ketone or an aldehyde or a carboxylic acid. And then you can generally will give you a carboxylic acid. They're slightly easier because you will also have a C to O group here, and you have the OH group, which is slightly further down about here. So I hope that makes sense. Next, in the chat, I was asked about describe precipitation reactions. Right, silver nitrate and a halide. So if you have that silver nitrate, which can split into... Now you just need something which can produce a halide, which is a halogen ion. Oh, one sec. When do you know to start your finger? Your finger. You'll never need to use a fingerprint region in this. Basically, the fingerprint region is anything less than 1000. That's the fingerprint region. But you'll never need to use it. Anyway, if you have something, let's say HCl, now that can split into H plus Cl minus. So you've now got a Cl, you've got your halide and you've got Ag plus. These can react together to form silver chloride. And now these, the ions, are aqueous, but this is a solid. And a solid which is suspended in aqueous solution is a precipitate. Now, what's the next part of the question? So that's basically the ionic equations for them. And you have bromine, iodine as well. Describe the use of precipitation reactions in tests for different halides. Right. Well, firstly, each you get different colors. So you have chlorine, bromine, and iodine ions. Now, in chlorine, you'll get white. You'll get cream if you have bromide. That's chloride, bromide, and iodide, you get yellow. So the precipitation test can be used to determine which one by their color. You can then also test them using NH3. If you have your silver chloride and you add dilute NHS, it is soluble. So soluble in dilute. This is soluble. Soluble in conc, and this is insoluble. So you can test, see which ones are soluble. And if it is soluble in dilute, you know you've got chloride. If it's soluble in concentrated, if you just try concentrated, it could be bromide or chloride. And if it's insoluble, then you've definitely got iodide. So I hope that makes sense. Okay. Why do you like my shirt so much? Right, where's the next question? That's all the questions I've been asked now. Right, ribosomes and Golgi. The important thing is just remember what they do. Ribosomes are for protein synthesis, and the Golgi is for packaging and processing protein. So basically, this is all talking about protein synthesis and kind of distributing whatever proteins you need. Now, let me just... All right. One sec. Sorry, one sec, I'm just checking something. I've never actually seen that question to do with neutrophils, because neutrophils tend to, the whole point of a neutrophil is so it can, well, it's a phagocyte, it'll digest things, so it needs to release powerful enzymes from the lysosomes, which is that main function. So I'm guessing ribosomes and Golgi are to produce these enzymes, the hydrolytic enzymes, which will then go to the which will then go to the lysosomes. That's what I believe. Hey, Steph. All right. Right, so dynamic equilibrium. That's from an AS. So normally it says equilibrium in AS. What specifically to do with dynamic equilibrium do you want to go over with, or over? Because do you actually want to know what dynamic equilibrium is, or do you want to know about effects of temperature and pressure? Also, one random thing, I am expecting in a few days' time, at the rate I'm currently getting views, I should be getting my 50,000th view in a few days' time, which is pretty awesome. What affects dynamic equilibrium? Well, there are three main things. Yeah, you have temp, pressure, conc, and concentration. Now, I'm going to just give the example they give in the book because it's easy. Let's also give minus 90 kilojoules. Right, so this is just one between, this is the heart of the process. Now what you look at with temperature, you look at the forward reaction. In this case, the forward reaction is exothermic. That means if temperature is increased, it's going to be giving more energy to the system, so it's going to want to give out less. If it's exothermic, it's giving out energy. So what it's going to do is it's going to want to take in that extra energy, so it's going to shift to the endothermic side. So it's going to shift that way. If temperature decreases, it's going to want to give out more heat, so it's going to shift to the exothermic side. Pressure is all about number of moles of gas. On this side, we have 4 moles of gas. This side, we have 2. So if the pressure increases, it's going to shift to the side, which has, I believe, less moles. I just need to check that. Yes, good. Or fewer moles, I should say, if we're using correct English. So as pressure increases, it's going to shift to that side because there are fewer moles of gas. If pressure decreases, shift that way because there are more moles of gas. Because as the pressure it squashes things in, so you want to have the same amount of same amount of atoms going around in the same volume. So if you decrease the moles, you get if you decrease the moles and the pressure's being increased, it kind of works out the same. Yes, you're allowed to post exam questions. Then concentration. Now, okay, this is just if you increase the product. If you increase the concentration of, let's say, the products, you'll get more reactant. So, because it will want to minimize the change in products. If you increase the reactant, I've done that wrong. Increase the reactant, you get more product. If you increase the product, you get more reactant. So just whatever you increase the concentration of, you get more of the other. So I hope that helps. Essentially, the things to look out for: number of moles of gas indicate pressure. Pressure increases, you go to the fewer moles of gas side. If it decreases, you go to the more moles of gas side. Look at the delta H. If it's negative and temperature increases, it shifts to the endothermic side. If its temperature decreases, it shifts to the exothermic side. And concentration, it just shifts to the other side. If you add concentration of 1, it shifts to the other side. I hope that makes sense. Unit 2, Module 2, how does a specific immune response work? Right. Right, specific immune system. Right, you've got a disease. A disease enters, the speed of disease. It's firstly generally eaten by macrophages such as phagocytes. You eat them. So they engulf it, that's through phagocytosis. And then they will present antigens on the cell membrane. That's the first step. You can also have just these, you can have it infect a cell, so nucleus has infected it, and they can also present antigens. And the other thing is it can just stay in the body with its antigens just being presented naturally. Now, once you've got these antigens being presented, you get clonal selection, and that's essentially when all these three will go over here to T helper cells, which will have corresponding antibodies on their cell membrane, T cells, they have corresponding receptors. So that will have on them, which will be complementary to the antigens. Now, sorry, I occasionally just read the questions. So those T helpers, but also you can have the infected cells and the pathogens themselves going to B cells. Now these have the antibodies on. That's an antibody which fits. And then these are found in the bone marrow. This is where the origin they are, reproduced. Because in the body you have thousands of different T cells and B cells, all with different receptors or antibodies on them. And then this clonal selection. So basically, the one with the correct antibody or receptor will bind to the antigens, and the body will go, right, this is the correct one. So you then get clonal expansion happening, where these turn into T killer cells, which will kill them. More T helper cells just increase the process, you get memory. T helper cells also stimulate B cells to divide, so you get more B cells, which some of them will remain as memory. The T killers will kill, and then lots of B cells will just release antibodies, which will then attach to the cells and cause them to be killed. It will kind of explain them. So that's step. So basically, a disease comes in. It infects cells and produces antigens. It's absorbed by phagocytes, which then present antigens, and it's also just in the body with its natural antigens. Now this causes clonal selection where T helper cells and B cells with corresponding receptors for T cells and antibodies for B cells are complementary to these diseases, so they kind of recognize it. Then they go through clonal expansion where they kind of replicate into T killers, which will kill them, T helper cells, which will act as memory in case you get infected again, stimulate more B cells to divide. which some of these cells will act as memory as well, and the others will release antibodies which will help kill the disease. So I hope that makes sense. Right. Can you please outline Module 3? You're going to have to be far more specific than that. Module 3 would take the whole revision session to go through, so be more specific, please. Why do we need 2 memory cell types? Well, if you have a T memory cell, it will quickly expand to form the T killers. If you have a B memory cell, it will quickly expand to form the antibody ones. So that's why a bunch of flowers may survive longer if the end of the stem is cut. It's to do with you have the newest cells. So the newest, because they'll cause cells to divide, and you get new cells, so they'll kind of be healthier. And also kind of a large surface area will be available. Right. The difference between literally T and B memory cells, one will just cause T killer cells to be made, the other will cause the B antibody cells to be made. Can you explain why the 4S subshell is present before the 3D cell? The subshells thing is just to do with energy levels. So right. It is the fact that the 4S subshell is at a lower energy level than the 3D subshell. And I'm not 100% sure why. There must be a reason. It just says here that the 4S subshell is just at a low energy level. That is the reason. Okay, that's the reason as well. They're in the xylem. It's to do with also, you also get new cells as well. Kind of a bit of both there. Yes, it is also about air bubbles. I forgot about that. Thank you for reminding me. But it's also, you do want new cells. New cells tend to be slightly healthy, and you get division as well. But yes, it is also air gaps. Right. Good. I hope that answered the chemistry question, by the way, about the subshells. Can I explain? Mohammed, what example do you do? Do you do OCR? Because I don't know how much detail I need to go in about transcription and translation at an AS level. I don't think you need to know about that much. Can you say if you're doing OCR, and if so, I'll try and answer it. Now, can I explain the process of evolution? Yes, I can, as I'm an evolutionary biologist, but at heart, yes, I can. Right. Well, the important thing is you have natural selection. Natural selection is the mechanism that drives evolution. I don't know what I'm going to my pen out. Again, can you transcription translation? Is that at an AS level? Because it comes up in A2 and it's. I don't know if it's at an AS level, but yeah. Oh, it is OCR. Wow. Well, I'll have to think about that, see the level. Anyway, evolution, basically natural selection. You get a random mutation So in the genetic code, a random mutation occurs, which could give a benefit to the organism. So let's say a long time ago, we were organisms without thumbs. And trust me, being without thumbs is very hard. And then there was one organism that got a mutation in its genetic code, causing almost like an extra finger being made where the thumb is. So it's kind of got the thumb where before there was none. Now that's going to help it survive. If it will help survive, it's more likely to pass on its genes. If it passes on its genes, it's going to pass on that gene for that good adaptation. And what happens over many years, you get the ones that are getting the thumb survive, and it becomes more refined over thousands upon millions of years until eventually there are organisms with thumbs and organisms without. A little bit. Yeah, I don't think you need to go into the full detail because it comes up in A2 in a lot more detail. I don't see how you need to know about mRNA and how it leaves the nucleus to go into nearby. Oh, okay, well that's all you need to know then. Yeah, it goes into a lot more detail at A2. Semi-conservative explanation. Right. You will not really need to know about semi-conservative because it kind of came up in the last June exam. So. Okay, wow, you really do not need to know much about transcription and translation. Right, semi-conservative. Did two people ask that? Yeah. Right. Well, semi-conservative DNA. Now, I'd say I don't think this is really done very well in the book. I don't follow as much on it at all. Oh no, people, right? Okay, I'm going to do this experiment. Right, as I say, it came up in my exam in June, so I doubt it will come up in yours. But basically, right. One sec. I've just. Oh God, all my books. 1 second. I've just dropped all my books, which is not very helpful. Sorry, 1 second. Right, well, okay, right. Sorry about that. Right, you have 3 types of theories of replication. There's conservative, there's semi-conservative, and there's dispersive. Now, the things you need to remember about semi-conservative is that it basically says you have DNA like this, it's 2 strands. Dun, 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 dun. Semi-conservative says this. You'll get, when it replicates, 1 strand will be new and 1 strand will be old. So basically half of it is new and half of it is old. Now, the experiment they did to prove this is they had a test tube with DNA containing that had started. They started their life with nitrogen 14, which is an isotope of nitrogen. It's a naturally occurring isotope. They then decided to add nucleotides that have nitrogen 15, which is a heavier isotope. Now, when DNA replicates, it forms a band. So this is the band of DNA forming. Now, they made sure that all the DNA already had nitrogen 14. All the nucleotides that were available, all of them were nitrogen 15. Now, if it was semi-conservative, what would you expect is to get one strand to be nitrogen 14, this one, and a new strand to be nitrogen 15. So if this is the starting point, you expect it, so that's the water or solution, you expect it to go lower by about half, and it did. The next time they did it, so you now have one strand with nitrogen 14, one with nitrogen 15. You will now expect one strand, the nitrogen 14 strand which is replicated, to have nitrogen 15 again. And they found that, so it was at that level again. But the new nitrogen 15 strand would also replicate, which means it would only have nitrogen 15. Which meant that one went even lower because it's even heavier. I've drawn that really badly, actually. One sec. Oh, God. I hope the explanation is being understandable. Right. No, essentially it was. Right. This is the top of the solution. Now you have a strand of nitrogen 14 DNA there. Then when it replicates, half is nitrogen 14, half it is nitrogen 15. So it's now heavier. Half of it is heavier. So you get a strand going down. It then replicates again. One strand of nitrogen 14 strand gets with nitrogen 15, so it's the exact same weight. So you get another strand there. But then you get another strand, the nitrogen 15 strand, also replicating just with nitrogen 15. So that becomes even heavier. So you get a lower strand. You need to know this. It's not in the syllabus exactly, so if it comes up on a paper, they have to explain it to you, and it came up in our paper, so yes, this can come up, this exact experiment, and they ask you to predict what happened. It will not come up in your paper because it came up in ours. Can you write down a step-by-step method? I can't write down the whole thing, it would take ages, sorry. If you need to, you just re-listen to the bit again or listen to it afterwards. Basically, you have all of this is nitrogen 14, then becomes nitrogen 14, nitrogen 15, half and half. Then you get nitrogen 14, nitrogen 15, half and half with a nitrogen 15 only strand. So I hope that makes sense. Please tell me if it doesn't. But yeah, it's not in the book, so it will not come up. I know it won't. I can promise you it won't come up. Can I predict what will come up in F212? I did that yesterday, actually. If you look at yesterday's live stream, you can have a look there. F322, I don't do OCR sorters, so I have no idea. Sorry. Oh, wait, F2. Oh, sorry, no, that was the thing with the wrong one. F322 is Unit 2. The answer with chemistry is everything. Everything could come up in chemistry. Also, what are the main things I should remember for chemistry? Everything. It literally is just everything. Because there's not much content, so... yeah, so just everything. mRNA and tRNA. Right. Nucleus with DNA. It unwinds and unzips. Yeah, coding strand. So this is at an AS level. And then mRNA starts joining to it. So you get a new strand, mRNA, which will then leave through the nucleus pore and go to a ribosome, attach, and tRNA molecules, which are shaped like this, carry. No, sorry, no, not like that. It's wrong. The wrong way around. Right, they carry an amino acid there and have 3 bases there, which can code to 3 bases here. And don't need to know. Don't ignore that. Right. So basically, mRNA is formed in the nucleus by kind of joining onto adjacent DNA, complementary DNA, goes to the ribosome, where tRNA molecules with an amino acid come and join, which are complementary to it. You have an anticodon, which is complementary to a codon. And to be honest, even that I think is a little bit too much. You need to know at an AS level. You just also, the main thing is to know is that mRNA is single-stranded, has U, not T, no T. Yeah, just remember it's single-stranded and has the base uracil, not thymine. And also it contains ribose sugar, not deoxyribose sugar, because it's RNA. That's all you need to know about that. Where are my questions? Difference between atrioventricular valves and semilunar valves. Okay. All right. Well, if you know the basic structure of the heart, if you just think about it, actually, the atrioventricular valves. Now they are the ones which are found in between, surprisingly, the atria and the ventricles. So on the right, this is a really inaccurate picture of the heart, but I can't draw an accurate one. So what are on the amino acids? What do you mean, what are on the amino acids? I don't understand the question. In the heart, yes, I know it's not what a heart looks like. Atria, ventricle. Now the atrioventricular valves are the ones here. The semilunar valves are the ones here that lead into the atria. Oh, sorry, no, wait, no, sorry, that's wrong. I can't look at my own pictures. Oh, God, I'm doing awfully tonight. They are in between the ventricles and the arteries that take away from it. So that's the pulmonary artery and the aorta. Sorry, making a few mistakes tonight. That's not very good. We're in New Peak. So the atrioventricular valves found between the atria and the ventricles. The semilunar valves are found between the ventricles and whichever blood vessel takes them away. And then, does that answer your question, as in the differences? That's literally what the difference is. They have the exact same function. It's just depending on where the blood is. So blood can flow through the atrioventricular valve. If it tries and pushes back, it will close. Then if it goes into the ventricles and then into here, that's awfully drawn. That's fine, but if it tries and gets back, the valves then again close. That's just all it is. Right. You need to know about where mRNA and tRNA are located, and you need to know about the single-stranded, the ribose sugar, and the uracil instead of thymine, but you don't need to know about the shape of tRNA yet. currently 22 now. Right. Explain tertiary structure reference. Right. I think I did this yesterday, actually. I did this yesterday. I will do it again, just quickly. Essentially, you have 4 types of bonds. You have hydrogen, which are between O and, so you have that kind of bond there. That's a hydrogen bond. Ionic, which are between charges. Disulfide, which is between sulfur and sulfur. I think it's that. And then hydrophobic philic interactions, which is just if they like water or dislike water. And basically, these different groups are found on the R of the amino acid, which we did probably in the nucleic acid video. Now, well, yeah, so if you have an oxygen on the R group of the amino acid, so let's say just it was CH2OH, that was it. That hydrogen there could then bond with an oxygen on another amino acid. So I'm just going to draw, actually, yeah, just ignore that. Basically, it'll bond. And then this will change to shape. So if you have, let's say, Na plus H, H, Cl minus, you will get a bond forming there, which could cause the shape of the molecule to be like that. So you kind of get a square. That's what affects it. So these will all cause bonds to form between the hydrophobic and hydrophilic, just depend on where. The hydrophobic R groups will go into the center of the molecule, and the hydrophilic will go onto the outside. And that's how it affects tertiary structure. No, I have no idea what time I predicted it, sorry. Step-by-step method in answering QWC questions in general. Any tips on how to tackle suggest? I did off. If you look at after an hour yesterday on my live stream, I did about how to answer exam questions. So if you have a look then in that one, so after about an hour yesterday, I did how to answer exam questions. Let's just have a look there. The dots just show a bond. Okay, so this is just a hydrogen bond. Okay. Do I know page 103? I don't know. Okay, is all the stuff I've done so far okay for everyone? Because I've kind of been a bit hectic and making a few mistakes. Sorry, I am correcting them as I'm going along. So is everything I've done so far okay before I continue on the next question, which is the glucose concentration one? Is that okay for everyone? Oh, and also, Mark, do you understand how to do a Benedicts test before I do the colorimeter? Do you want to go through that as well? Okay. Right. Well, firstly, the colorimeter question. Actually, no. I'll leave the colorimeter because I just want to see if you actually know how to do a Benedict's test first. Right. Next one. Can you explain in situ and ex situ? Right. Well, in situ just means keeping it in its habitat, natural habitat. I think if you want to do that, it's like law. So outlaw poaching, really make sure you stop poaching. Create national parks so people can't hunt in those areas. Make sure there's no deforestation or getting rid of the habitat. You have to make sure that you've got, a good area which has all the relative, food, water, shelter, space for all the organisms that want to live there. You can repopulate it. So you put animals back in. You can have reserves, so even a healthy natural environment, a secure environmental future, sustainable use of the environment. But basically, you want to make sure that you stop hunting. All the materials are there, there, so they've got all their food space. Mark, can you quickly go through that? Is that the Benedict's test? Just say yes to that, and I'll do that next. But yeah, so you want to have, keep them in a natural environment and stop hunting, poaching, deforestation, or removal of habitat, and make sure they've got all the things they need. Exit is when you get things out it. So you've got biological kind of centers. So you've got seed banks for plants. So you can store seeds, freeze them, and then grow them when they are needed. You've got zoos for animals. So you can put animals in a zoo and breed them together and then release these animals back into the wild. You can educate people. And yeah, you can also freeze sperm, which will so you can use that again later or give it into animals. Research animals. And also you can save if a species is about to die, you can put them in a zoo and they'll still be alive. Okay, right. Just quick, before I hope that answers your question, whoever asked about in situ and ex situ. State the MZ value of the molecular ion peak. Okay, what does that even mean? Very quickly, this is a very simple one to answer. On a mass spec peak, the M plus value, the MZ value, is just look at the peak, which is the furthest to the left. So if you have, let's say that says 104. Right, you look for this peak, the furthest along, and it's that value, 104. That's all it means. And that is equal to the MR of the molecule. Right, Benedict's test. Right. Now, Benedict's test, you just want to... Now, Benedict's test tests for non-reducing sugars. And reducing sugars. They test for both, actually. I'm sorry, but we're going to look at reducing sugars. Glucose is a reducing sugar. Yeah. Basically, you add Benedict's test. So add Benedict's solution. Heat to 80 degrees Celsius. And what you'll see is a change in colour from blue to green to yellow, orange, and then finally stopping at brick red. So that's what you do. Just add Benedict's and heat it to 80 degrees. Right. Okay. I'm sorry. I need to say this. Right. Have I included this in the video? Everything in AS has been done. Every single topic in the book at AS is covered in a video. So, yeah, it is done in a video. Yes. So anyway, once you've done this test, now what you want to do is you want to perform this test on your unknown sample. So perform it on your unknown. Then what you do is you perform it on a set number of known samples. So let's say you'll try it. I'm just draw it. You do it on 0 mol per dm cubed, 0.2, 0.4, 0.6, 0.8, and 1.0. That's the concentration. And then what you do is you look at, because what happens, a colorimeter will shine a beam of light through a sample. So I should have mentioned it. And a photoelectric cell picks up the light that's passed through the sample and gives you a reading about how much light has passed through. So you look at the different concentrations, how much has been transmitted through. And it says here, as glucose concentration increases, it's more is transmitted through. And then what you do is you test your value and you see how much is transmitted. So let's say that much was transmitted there. Draw a line along, and then you draw a line down, and here you know that your sample is probably 0.5 mol per dm cubed. So basically shine light through it, and yeah, you just shine some light through it of known samples, see the percentage transmission, or yeah, percentage transmission. You draw a graph, and this is a calibration curve. It's calibrating it kind of. Then you try your own sample, see how much is transmitted, do a line across on your graph, a line down, and that's roughly where the concentration will be. Does that make sense? Right, EZ isomers. All right, let's say you've got the molecule, have a carbon double bond, so you've got now this is it's butene. Where you draw it, you draw the carbon with a double bond, and then you just draw the groups. H, you draw the groups, and then when you draw your second one, you just flip it around. So you got here, we got them on the same side. On the other molecule, we had them on opposite sides. Right. Immunity part, can we say antibody antigen complex? I've never seen that written. I can't see why you couldn't say it, but I've not personally seen that written. I think that'll be fine because that is what it's forming. So yeah, I think that'll be fine. Cofactors and coenzymes. Right. Where's that? Right. A cofactor is any substance that must be present to ensure enzyme control reactions take place. They are part of the enzyme, either prosthetic groups, or they can be a temporary basis, which are coenzymes. So you have cofactors is the general word for anything which is bound to an enzyme to make it work. And there are two main types. There's coenzymes and prosthetic groups. I'm going to do prosthetic groups first. Let's say you have hemoglobin. I'm going to draw just that. The prosthetic group will be here. That is heme. I spelled that right. Please someone correct me if I spelled that wrong, because I think I have. Is it HE? Yeah, it is. Sorry. I'm very sorry I spelled that wrong. The heme group. Oh, sorry, go back. That's probably do all these. What right? Because let's say you had propene. Like that. If you look at the structure, right, this can't show cis and trans isomerism because there is the same group on one carbon. So you've got two hydrogens on the same carbon. So if they were to swap around, so if you broke the bond here and swapped them around, you had CH3H here, it's the exact same molecule. It's not got a different arrangement in space because you can just flip the molecule upside down. Yeah, I know my handwriting is awful, especially when I'm trying to write it quickly. Sorry. Everyone who watches my videos should know by now that I work by speaking, not by writing down. Writing down is just notes. Yeah, it can't be described because some have the same group, two of the same group on the carbon. Right. Anyway, back to proteins. Prosthetic group, that is something that is permanently attached and will have the effect. So this is the iron in it. There. That's what has the effect. Now, a coenzyme is something that perms. There is small organic non-protein, it's always non-protein, and binds to a short period of time. That's all it is. So if you have a molecule here, that's our protein. What you could have this joined. We'll change the shape. Let's say change the shape slightly by doing that, just binding there. And by doing that, that opens up the site. So if, let's say, the substrate can bind to it. So that's what a coenzyme is, something that partially binds, not always. And a prosthetic group is something that permanently binds. A cofactor is just anything that's like a coenzyme or a prosthetic group, just something that binds for a while to it. Do I get paid? No. If that is genuinely to me, no, I do not get paid. I've not got a single penny out of doing this. Where are we now? Jessica, can I explain blood, tissue, fluid, and lymph? I believe I did that on 2 nights ago on the live stream then. Equally same with CJ1369. I did discuss the structure of collagen on is it Saturday? Yeah, Saturday night's live stream. So if you look at that again, Saturday's night's live stream, I've both described the structure of collagen and blood, tissue, fluid, and lymph. And one thing I said is that the structure of collagen you will not be asked in your exam because there's a 6-mark question on the previous exam on it. So I doubt you'll be asked. No, coenzyme is not a coenzyme. Is not a protein. It is a non-protein group that binds to it. So, for example, if it it could be something like zinc. I think zinc is a prosthetic group, but it's just something like it's not a protein. And proteins are something that just have the a protein is made up of those, the amino acids. If it doesn't made up of amino acids, then it's non-protein, and that's a coenzyme if it binds to help. Do we need to know how carbon monoxide causes atherosclerosis? Let me check. I'm pretty sure you do. Yes, you do. The way that carbon monoxide, it damages the inner lining, the endothelium of the arteries. If the person has high blood pressure, which carbon monoxide also leads to, is high blood pressure, it will add to such damage. Now the damage is then repaired by phagocytes, which encourages the growth of smooth muscle and the deposition of fatty acids. The deposits include cholesterol from low-density lipoproteins. And when you do this, these balls of fats are called atheromas. I hate this word, atheromas, which can then cause, blockages. Right. Why is genetic diversity important? Because if you don't, you can get, let's say you have a group of plants which all have very similar genes and a disease comes, none of them will have resistance to it, so the whole group will get wiped out. With genetic diversity, you get mutations, you get differences, you get adaptations, which means some will be able to survive when others won't. If there is a disease or a natural problem, which is a problem, well, dangerous, then having genetic diversity can mean that at least some organisms survive. And did I say epithelium? If so, I'm incredibly sorry. I meant endothelium. I even saw it written down. God, these things are really tiring. That's why. Can we do past papers in these sessions? Maybe. I'm posting a video tomorrow about future live streams and what people want to do, so we can discuss it then. Genetic diversity, coenzymes, done that. Explain what we need to know about seed banks. Is that, well, they exist. That's always a good thing. Just need to know that there's a thing called a seed bank, and that they store seeds, and how they store seeds and why they store seeds. What they do is these seeds can remain viable for decades, which means if a plant dies or you need a sudden, the plant's coming endangered, you can regrow these seeds. And you also test them and see what makes them work, what makes them not work. Now they're stored in very dry or even freezing conditions, and they're resistant to desiccation. The level of moisture in each has a direct effect on storage. For every 1% decrease in seed moisture, the lifespan doubles. For every 5 degrees reduction in temperature, the lifespan also doubles. So if you have it really cold and really no moisture at all, it's going to live a long time. And you need to know about how they carry out germination tests. So they remove samples and germinate them periodically. They plant the seeds in petri dishes of nutrient agar, so agar plates, and keep them in controlled conditions. They measure the germination rate and the success of germination, and this enables scientists to monitor the condition of the stored seeds. So that's what you need to know about seed banks. Mark, no, because I don't really know. I didn't do the F211 exam last summer, so I don't know what was on it, so I can't. Sorry. Right. Any more questions? Well, I think I've answered all the questions that I've got the backlog of. Right, just don't ask any questions right yet. Just can everyone who is watching and has asked for various things, can you just comment if all the stuff I've said in this video has made sense? It all, it's helping you, just so I know that I'm doing well in this video. No, I do not remember everything by literally reading out continuously everything I wrote. No, I don't do that. I make notes, which I'm reasonably good at remembering them, and then just practice, and anything I get wrong, I reread and redo. How's a hydrogen bond formed? CJ, is that in terms of biology or chemistry? Because chemistry goes into more detail. Actually, I don't read my notes because I don't like reading my notes and they're messy anyway. What's the C14? Sorry, Mark. What's C14? Oh, that's nitrogen, not carbon. Oh yeah, one thing in F211, I'm expecting a translocation question on movement of sugars. That hasn't come up for ages. The three test tubes. Mark, I'm going to say don't worry about that. It will not come up in your exam. It came up in my exam, and it's very rare that it ever does come up. So it will not come up in your exam. Do not worry. Fine. Okay. Um, well, that makes it a bit easier actually, because I don't think you need to know it in that much detail. Right. Well, basically, water is charged. It has these things called dipoles, so it has charges. These are slight positive charges or a slight negative charge. And what happens is, it means that, as you know, opposite charges attract. So the positive and a negative charge can form a hydrogen bond. So you get hydrogen bonds forming because there's a slight positive charge on the hydrogen, slight negative on the oxygen, and oxygen and hydrogens on opposite water attract. That's what you need to know. No, I don't read my notes at all because I've written them. If I need to, I read things from the official text. My notes are made from all the different books, so I use the books to read from. So I read from the books, and I just do lots of practice. Practice is so good. I'll do hundreds upon hundreds of questions, and then see what I haven't got wrong, and then go. Right, Jessica, you'll get 2 8 or 9 markers in F212. In F211, you won't get anything that big. You'll get 2 6 markers, 5 or 6 markers, maybe a 7 maximum, but not much bigger. What do fertilizers, pesticides, and antibiotics do on improving production of food? Basically, no, it's gone. What they do is pests. So pests like insects will and diseases, bacterial diseases, will kill plants. If you have 100 plants and 50 of them are killed, you've lost 50% of your yield. Pesticides and antibiotics will, particularly antibiotics are more on the animals, sorry, but pesticides, let's say, out of plants, if you do them, you might only lose 10 plants out of 100, so you've instantly reduced your yield. Fertilizers give nitrates into the soil, which encourage plant growth. Plants need nitrate to live. More fertilizers give more nitrate, which help them to grow. Antibiotics are like pesticides, but for animals. So what we'll do is they'll cure them of diseases, so they need to spend less energy in fighting off the disease, so they can spend more energy on growing. That's what you need those ones for. I hope that answers your question. It depends. If it says, if it's a 2-mark question, so it wants 2 points and you give 3, it will mark the 2 correct ones as long as the wrong one doesn't contradict the other one. If it says state 2 points, says, so, you know, it says 2 things in, you mentioned 3, it could mark you as wrong because you've mentioned 2 things for 1 point. So that could, so it depends. Let's say it just was a 2-mark question, so you expected to write 2 points. So if you have to write 2 points and you write, let's say, what is the function of, I think of a good one. I can't think. I'm terrible at thinking up questions. If it says state 2 points, let's say it said what I'm doing maths example. Let's say what numbers equal 4, and you said 1 plus 3, 2 plus 2, and then you said you then wrote 2 minus 2. Well, you've contradicted yourself because you said 2 plus 2 equals 4, but so does 2 minus 2. That's a contradiction. And since that, you've made that one wrong as well. You've made that one null and void. So as long as you don't contradict yourself and you say, and it doesn't state specifically for 2 points, then you'll be fine. You won't lose marks getting an extra wrong point. Are there any topics that you think might come up in F2128 markers? It's stuff like water, how water works, that's always a very good one. The immune system, that's a very common one. Things on biodiversity, so they ask like reasons for maintaining biodiversity is a bit, or conservation, big marks there. Something on food, they can ask a big question on food. Generally the big questions do tend to come from module 2 and module 3, except if it's water, and water's the only one from module 1 which they ever asked, which is big. No, you don't say to look at the last answers. No, James, they will mark. Examiners want to give you marks. So as long as you haven't contradicted yourself and you've made 2 correct, because there are 2 types of incorrect mark they can give, and that's the ignore mark and the reject mark. Now, reject marks only appear if you have to have stated, if that's, it's a 1 marker, so you have to get it right, and you've got it wrong. That means reject, you've got it wrong. Or if it's because you contradicted yourself, or if it's because it says, if it says, state 2 points and it said you've done 3, it says if additional point is added, reject that whole line. Ignore marks mean you've got it wrong, but it doesn't matter. We're not going to take a mark off. We're just going to ignore that statement. So if you say, let's say it was a 3-mark question, and you say 4 good points, correct points, and 1 incorrect one, they will ignore the incorrect one because you've already got the 3 correct marks. Oh yes, DNA replication, of course, I forgot about that. Yes, you can get a long one on DNA replication. Yes, you do need to know about the roles of the Casparian strip and the transpiration stream and pull. Yes, you do. No, it means just ignore that 1 point. So if you've made 10 points and one of them's wrong and it says ignore, you just ignored that 1 incorrect point. They don't do it. They just act like it wasn't there. Any big markers? Oh, there could be anything. Usually a big marker on identification of a molecule. Let's see what else is there in here. The calculation could be a big one. They might ask for, lots of something to do with a reaction pathway and a mechanism to go with it. Yeah, it could be a few. All the functional groups you need to know. You need to know about the alkane group, the alkene group, the alcohol group, the halogeno-alkane group, where X equals halogen. Let's see. You need to know about the aldehyde group, the ketone group, the ester group, and I think that's most of them. I don't think of any more I need to add. Yeah, I think these are all the functional groups you need to know. So alkane, alkene, alcohol. Oh, another one, carboxylic acid. There you go. So alkane, alkene, alcohol, halogenoalkane, aldehyde, ketone, ester, and carboxylic acid. Yes, you do need to know about transmission, malaria, HIV, and tuberculosis. Do we need to know about the potometer? Yes, you do. It came up in the January paper last year, so again, I can say that probably won't come up. But potometers, yes, you do need to know about them, particularly about features of how potometers work. Cell signaling, that's a classic. Now, all cell signaling, they make it to be such a big thing in the book, but there's essentially just one thing you need to know about it. It's just really one, and that's the definition. Cell signaling is the communication between cells using chemical messengers. That's all you need to know. A C-double-bond-C is a functional group. It's what's responsible for the molecules. So yes, it is a functional group. What was I saying? Yeah, so cell signaling is just basically communication between cells using chemical messengers. And just know that basically, you need a complementary shape between the receptor on the cell and the molecule. If you want to know what I got on the practicals, in AS, I'll write it down for you, because this is one bad thing with the practicals, is your mark will go down due to grade boundaries. Bio, I got, that was raw, but it changed to 50 out of 60. Chem, I got 35 out of 40, which changed to 48 out of 60. A2, bio, I got raw 39 out of 40. Chem, I don't know yet, but so far I got 14 out of 15 on the quantitative and 7 out of 10 on the qualitative. Yeah, those are my practical results so far. Whoop. I'm interested, which practical did you lose a mark on? Because I can tell you which one I lost my 1 mark on for the bio, and I'll tell you why I lost it. Just tell me which one you lost your mark on, and do you know why you lost that 1 mark? Because I know why I lost my 1 mark, and it's the most stupid reason ever. QQE, is that? Oh, the practicals. Yeah, the qualitatives were ridiculous, and the quantitative had a rubbish mark on it as well. There's one quantitative where one of the marks you had to go to the nearest second on time, yet they never said that. So on the stopwatch, my whole class, out of all 3 of us, did it to 2 decimal places like it is on the stopwatch, but we all lost that 1 mark, and that was the only mark I lost in that quantitative because we had to do it to nearest second. Yet it said nowhere on the paper, and my teacher didn't even know why they said that. Yeah, it's converted to it's converted to UMS. The raw mark is 37 out of 40. That's the UMS, the mark I got overall. The actual one that's on my exam certificate. Evaluative. Okay. I lost mine on the qualitative because I didn't draw a board around one of my tables, and I lost a mark for that. Which I don't see how that benefits my A-level biology at all, kind of drawing a board around a table, but apparently it does enough to lose 1 mark. My teacher just did because she got so annoyed with the fact that I lost that mark. She was like, she thought it was a ridiculous place to lose a mark. James, no, it isn't cheating as long as we don't specify exact questions. I've not said a single question that's been on a paper, so don't worry. As long as we don't say what's on the exam, it's fine. Yeah, it's a thing because yeah, which I know it is ridiculous. I literally lost a whole 10% because of that. Um, yeah, it is ridiculous to grade back. It's because so many people get around 36, 37, it does end up going down. What I'm annoyed about is when teachers end up telling students the answers. That's really annoying. My chemistry teacher refuses to tell us a single thing. He'll barely tell us the title of it. He'll literally go, "Yeah, you need to learn this, right? Go," and that's it. Our biology teacher will tell us and then tell us to kind of literally go, "Look, research this, learn everything you can on this topic." And she won't tell us the questions. She'll go, revise this topic. So yeah. Best thing to do before an exam. It depends what sort of person you are. I suggest just do some practice, go through stuff, anything you don't know, go over. But what you want to do is you want to do stuff that will make you confident. You do not want to, do a paper and go, oh my God, I know, only know half of this, I'm going to fail. You don't want to do that. But I personally, on the day before I will revise. On the day of an exam, particularly the exams in the morning, I do like to relax. Just so I'm nice and calm before the exam. Yeah, well, they are changing it. When the new A levels come in, practical coursework is gone. There's no more practical coursework. It's 100% exam based from I think 2015. So the year that's starting this year will get our same exams, but the year starting next year, so the ones who are currently in year 10 will have no practical exams, no coursework, so they won't be able to cheat like this. Okay. As chemistry, clarify and simplify how fractional distillation of crude oil works. God, I haven't done. Right, fractional distillation basically works because different lengths of hydrocarbons have different boiling points. The longer the length, the higher the boiling point. And basically, you put crude oil in and you heat it to a very high temperature, and which kind of makes everything into a gas. You then put it into the column, which is very hot, a high temperature at the bottom and a lower temperature at the top, which means gas obviously rises. So as gas rises, anything which then has a temperature below its boiling point will turn into a liquid and can be kind of separated. So if you've got perhaps petroleum, which has the highest, the lowest boiling point, that will be a gas and rise right to top and leave maybe as a gas, or yeah, it will leave as a gas. And then something like diesel oil, that'll have a boiling temperature much higher. So it's going to halfway up the tower, perhaps it will then start to liquefy and condense and be able to go off. No, not new student. No, no. The A2 students will be, and also the new AS. It's the current year 10s when they start at AS will not be doing practicals. So they've got a whole year as well. Ezra, you really know we've all had to work to do them. You shouldn't just get the answers. And to be honest, it won't help. Knowing the answers actually does no help at all. Also, one thing, remember, you are asking to do this on a reasonably public area, which makes it quite obvious. Just saying. All right. Out of interest now, does anyone have actually any questions on AS biology or chemistry? Just any more? So I can answer. If not, I will head off. I'm going to leave at 7, so ask any questions if you have any, and I'll try and answer them in the next 12 minutes. Yep, feel free to ask a quick A2 question. Go ahead. What's my plans for this afternoon? What do you mean? Considering it's basically evening, the afternoon hasn't gone. What do you mean? What's my plans? Okay, see you, Mark. I've done brown like brownie. I did chlorination in was it? Yeah, I think I did that in Saturday's video. I've done chlorination already. So if you look in that video, I've done it there. Right. The right. Oh, this evening, no, I'm not going to be doing any more revision. I'm going to relax because these actually these live streams are so tiring on me. Right, everything on card isn't pumped into the intermembrane space. It stays in the matrix. It doesn't actually stay in the matrix. There's a good picture of it on page 90. What happens is you get the 2H from the Krebs cycle, so you get hydrogen atoms, which will give electrons to FAD, so we actually have the effect on FAD. But the hydrogen ions are then just pumped through in a different electron carrier, or yeah, so they might go through protein number 3 or 1. There's mass. Yes, you do need to know the mass flow experiment. I've never seen it come up. You could, yeah, you could be asked. But yeah, no, the hydrogen ion. So the hydrogens will be have their electrons removed and given to FAD, which goes through that electron carrier. But the hydrogen ions can then go to through other electron carrier proteins. Procedures and requirements. Procedures is applied through UCAS, just like any other university. Requirements is generally 3As. In biology, preferably chemistry and anything else, if you're doing biology. That is, if you're not doing biology, biology isn't a requirement. But for biology, as what I'm planning to do, it is 3As for biology, preferably chemistry, which I'm doing, and one other subject. Not a huge amount of preference, as long as it's a good subject. They will not count general studies or critical thinking. What's the experiment? Oh dear. Actually, yeah, where's the mass flow experiment? Actually, no, I must be wrong. The mass flow experiment, is it in the textbook? If so, what page is it on? Because I can't see it, so I think since it's not in the textbook, you don't need to know the mass flow experiment. Hello. For biology, you don't need work experience, but it always helps. So I've worked at the Cat Protection League and also at the East Sussex Wildlife Rescue and Ambulance Service, so working with both domestic animals at the Cat Protection League and wild animals at the East Sussex Wildlife Rescue and Ambulance Service. And that definitely helps. Having experience does help. You do not need it, but it does really help. I would like to go into teaching. Obviously, A-level biology teaching, or depending on how much I like it, university lecturing / research. But I think probably I'll end up going to A-level biology teaching. Yes, it is in the revision guide. You're right, it is in the revision guide, which I haven't got with me, so I can't go through it. But I don't think you need to know it personally. Well, if it does come up in the exam, they would explain it to you. Since it's not really part of the specification, they would explain it. Well, page 81, but with other experiments. Well, on page 81 it shows evidence. It doesn't show the actual experiment. I know which the experiment that Mohammed is thinking about, but no, it isn't. It isn't in the text of that experiment. I don't know when it's high affinity or low affinity. Basically, the further the graph is to the left, the higher the affinity. That's essentially what it is. The higher the graph is to the left, the higher the affinity. Okay, and bye, James. Right, well, I think everyone seems to have asked all their questions now. Yep, right, so I think it is time for me to head off now. So, thank you all again for watching. As I said, I will post a video tomorrow on basically getting you guys to comment whether you want more live streams and what you want, and whether you want me to go through a paper or something like that. If so, which papers, or if you want me to go through more material, which one you want. Like that. So I'll go through that, and dates and times and things like that. I'll go through it tomorrow. In terms of videos, I'm now a little bit behind because I was meant to do a video today, but honestly, I just couldn't be bothered, really. They are quite a lot of effort. But it's over the next week and a bit, I will be doing chemistry, trying to finish off the A2 chemistry videos. And the A2 biology will be finished as soon as I learn that final module. I'm predicting within 3 weeks at the start of term, hopefully. I will get them done in plenty of time. So thank you again for watching, and I will see you in the video tomorrow to discuss more live streams. So thank you, everyone, and I'm going to say goodbye and just wait a little bit in case anyone wants to say anything just before I go. Also, try to watch as many of my videos as you can over the next day or two. See if we can get up to 50,000 views. That'd be pretty awesome. Okay, that seems good. So I will see you, say tomorrow. So thank you and goodbye.